we're going to start talking about cellular processes, which means we're going to talk about how cells maintain homeostasis and how they get the energy to do so. It's exciting. As we know, all living things on this planet are made of cells, either one cell or a collection of cells. We also know that all living things on this planet have a certain set of characteristics. We know that they respond to stimulus, they have DNA, they grow and develop, they reproduce. There was two other characteristics of life that we're going to be talking about in detail in this unit. We're going to be talking about how cells maintain homeostasis, how they maintain a balance inside of themselves, and also how they metabolize food, which means how do they break food down or they get energy to maintain homeostasis. Those are cellular processes. There's a law of the universe called the law of entropy. That means that things have a natural tendency in the universe to go from a state of order to a state of disorder. You've seen this law at work in diffusion. So if I was to spray a bottle of perfume on one side of the room, eventually people even on the other side of the room would be able to smell it. Because of the random movement of particles in the air, the concentration of the perfume would go from a high concentration on one side of the room to a low concentration spread out evenly all over the room. It's going from a state of order to a state of disorder. That's the natural tendency of the universe, and we say that's going with the concentration gradient. So that would be like if I had a beautiful tower of rocks, it wouldn't take long for me to tap it and have all of those rocks spread out all over a stream bed. However, if I was to build a beautiful tower of rocks, it would take a whole lot of energy for me to get against the natural law of the universe, to go from a state of disorder to a state of order. So if we're trying to go against the concentration gradient, then it's going to take energy. So, if you look at those guys in the bottom left-hand corner of the screen, you'll see that you've got a couple of guys who are hanging out, just going with the natural laws of the universe. There's a hole in the boat. The water's going from a high concentration to a low concentration inside the boat. That doesn't take any energy. However, you see on the other side of the boat, there's a couple of guys putting a lot of energy to go against the concentration gradient or go against the natural movement of particles. So they're going from a low concentration inside the boat to a high concentration outside the boat. And look at them sweat. Instead of water and things moving in and out of a boat, let's talk about how water and other nutrients move in and out of your cell. In order to do that, we have to talk about the most important organelle that protects your cell and lets some things enter and some things leave. It's called a semi-permeable membrane, which is because it monitors things so closely and makes sure that only certain things can enter and leave the cell. The membrane is made out of a phospholipid bilayer. It has a phosphate head and two lipid tails. Now the phosphate head likes water, so we call it hydrophilic. The lipid tails are fats, and we know that fats do not like water. But fats like fats. You can mix two oils pretty easily. So if I was just to let these guys go and hang out in the middle of an ocean, let's say, the fatty tails would actually be attracted to each other and running away from the water. And so they'd end up sticking together in a bilayer or two layers with the phosphate heads poking out towards the water and the fatty tails hiding from the water inside of the membrane. The thing is that this type of membrane doesn't stay still. It's not what we call static. The cell membrane is constantly moving, so we call it a fluid model. In this case, we actually call it a fluid mosaic, and that's because it's not just made of phospholipids. It has all sorts of things that are stuck in the cell membrane. It has proteins and sugars and cholesterols, all sorts of things to keep that cell membrane happy and healthy and to help it do what it needs to do. So let's think about some things that the cell membrane needs to do. It needs to help communicate with things outside of the cell and get those messages inside of the cell so the cell knows what its job is and what it needs to do. It also needs to let things in and out as they're needed. The last thing that the cell needs to do is it needs to let the body know that it's happy and healthy and that it's where it's supposed to be. So there are different communication proteins, sometimes called receptor proteins, that help the cell communicate. But we're going to be talking about two different types of proteins. One is a channel protein and one is a pump protein. Now, a channel protein is just like those toys that you played with when you were a kid, where you had to take a shape, a star or a heart, and then find that shape inside the little ball, and you'd push the shape through. But it would only go through one special hole on the ball, and that the shape of the hole had to match the shape of the shape. Channel proteins are just like that. They go with the concentration gradient, and if you remember, that doesn't take much energy at all. But they only let certain molecules in. There's one channel protein called an aquaporin. The aquaporin is an aquapore, or it's a channel for water to move in and out of the cell. Another important type of protein we'll be talking about are protein pumps. Protein pumps take energy because they go against the concentration gradient. And as they open, they get what they need, and then they close, and they let what that needed item is into the cell. Now, let's watch a brief movie on the cell membrane.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Osmosis is the diffusion of water across the cell membrane, aided by aquaporins. Because of those aquaporins, the water is able to move easily in and out of the cell membrane, even though the phospholipid bilayer doesn't like water very much. Tonicity is probably a word you haven't heard before. Tonic refers to a solution with dissolved salts and sugars. So tonicity talks about how much dissolved stuff, or dissolved salts and sugars, are inside of a solution. There are 3 ways that we describe tonicity. Hypertonic means that something has more salts and sugars. Hypotonic means that it has less salts and sugars. And isotonic means when you're comparing 2 things that they have the same amount of salts and sugars dissolved in solution. Why is this important? Well, it's important for homeostasis. Because water moves freely across the cell membrane, water moves from a high concentration to a low concentration. But if you have more dissolved salts and sugars, then you have less water. And so if you put a cell in a hypertonic solution, a solution that has lots of salts and sugars. See all those dots? Those are the dissolved things. The water is going to move out of the cell because it's going to go from a high concentration in the cell to a low concentration out of the cell. I have a phrase. The water moves towards the dots. The water always moves towards the salts and the sugars. So if you remember that phrase, you'll always remember the direction of the water movement. In a hypotonic solution, which is on the left, which is right here, In a hypotonic solution, you have a lot more water on the outside of the cell than in the inside of the cell. So, you have to think of the cell is hypertonic to the solution, and the solution is hypotonic to the cell. So when you think of it that way, you put the dots where the more solutes are, and in this case it would be inside the cell, and then you draw your arrow of the water movement towards the dots. In a hypotonic solution, that would mean that the cell is hypertonic, the cell has more dots, and the water moves into the cell. In an isotonic solution, you're going to see equal water movement into and out of the cell because there's an equal amount of salts and sugars and an equal amount of water. And for an animal cell, that's really good because it's going to help it maintain homeostasis. Osmosis is the movement of water across the cell membrane from a high concentration to a low concentration. That means, in other words, I could say it's the diffusion of water across the cell membrane, and it's aided by that protein called an aquaporin. Tonicity doesn't talk about the water as much as it refers to the numbers of salts and sugars that are dissolved in a solution. So if a solution is hypertonic, then it has more salts and sugars. If a solution is hypotonic, then it has less salts and sugars. And if a solution is isotonic, then it has an equal number of salts and sugars. Iso means equal, hyper means more, and hypo means less. In the diagram below, you can see the first diagram shows you an isotonic solution, where the solution inside of the cell has the same number of salts and sugars as outside of the cell. Because they have the same numbers of salts and sugars, they also have the same amount of water, which means the water is going to move equally into and outside of the cell, freely moving through those aquaporins. In a hypotonic solution, there's a lot less sugars and salts outside of the cell, which means that there's more sugars and salts inside of the cell. I always try to figure out where there's more sugars and salts, and then I draw in dots to represent the sugars and salts. I then draw a big arrow going from towards the direction of the dots. So remember that water always moves towards salts and sugars. So wherever you can put the most dots is where the water is going to move towards. In that second box, in a hypotonic solution, that tells me that the solution has less salts and sugars than the cell. So I put a whole bunch of dots inside that cell, and then I draw a big arrow in because water moves towards the dots. In the next set of conditions, it's in a hypertonic solution. Remember, hypertonic means more sugars and salts, which means that the cell has less, which means I'm going to draw my arrow from inside of the cell to outside of the cell in the solution. Although water always moves towards the dots, it can affect different types of cells differently. So if you look at the animal cell across the top, in a hypotonic solution where there's less salts and sugars outside, it means it's hypertonic on the inside. So there's tons of salts and sugars on the inside, the water moves towards the dots and into the cell. But the animal cell is going to expand and expand and expand until it bursts, or scientifically we call that lysing. In an isotonic solution where you have an equal movement into and out of the cell stays nice and balanced and is happy. In a hypertonic solution, an animal cell is going to still lose its water. Remember, hypertonic, more dots in the solution, water moves out of the cell and towards the dots. The cell is going to shrivel, and that's not very good for the cell. The cell becomes dehydrated. In a plant cell, it's got a different set of organelles. If you remember correctly, it has that special large central vacuole and a rigid cell wall. That cell wall acts like a box that's around the cell. And those boxes are stacked up to make a plant stand up straight. Remember, they don't have a skeleton, so they need those boxes to be nice and strong. Well, when the vacuole has water move into it, it expands and it pushes against the box on the outside and it makes the box really rigid. Like if I took a water balloon and I filled it up with water and then I shoved it into a shoebox and I tried to push down on the shoebox would be more rigid than one that did not have a water balloon inside. So in a hypotonic solution, when the water is moving fast into the cell, then that's really good for a plant cell because it makes it nice and rigid. We call that turgid. In an isotonic solution, you have the equal amount of water moving in and the equal amount of water moving out. But now that's like the water balloon's not really full inside of the shoebox. And so the shoebox isn't as hard and it's going to make the plant start to wilt. We call that flaccid. In a hypertonic solution, the water is still going to move out of the cell, but in this case, when it moves, the vacuole completely collapses and the cell membrane pulls away from the cell wall. And that's what happens when a plant loses so much water that even if you water it, it's not ever going to recover. We call that a plasmolysed cell. Below me, you can see in a hypertonic solution, the plant cell has been plasmolysed, and you can actually see the cell membrane has been pulled off of the cell wall. So far, we've learned about two types of passive transport. We've talked about diffusion, where things wiggle across the cell membrane, and facilitated diffusion, like osmosis, where an aquaporin creates a channel to help the water move across the membrane. Now we're going to learn about active transport. There are two types of active transport. There's bulk transport and there are protein pumps. Bulk transport involves particles that are too large for them to move easily through the cell membrane or through a channel. In that case, the cell membrane envelops the large particles and takes the entire chunk inside the cell. Inside the cell, it fuses with the lysosome. The lysosome, if you remember, is the recycling center of the cell, and so it releases its digestive enzymes and breaks down those pieces until they can be recycled. Whatever's left and the cell doesn't want is shipped back out of the cell. When stuff is shipped into the cell, we call it endocytosis, and when stuff is pushed out of the cytoplasm, we call it exocytosis. But because this is such a heavy involvement of the cell, it takes lots of energy. Protein pumps also take energy. They use the energy in ATP, our energy molecule, to go from a low concentration to a high concentration, which means it's going against the concentration gradient. So if a cell wants to move something against the concentration gradient, then it is going to have to take energy. That is active transport. The cell needs to move things in and out of it so that it can maintain homeostasis. Remember that maintaining homeostasis for the cell is hard work. Sometimes it takes energy to help it maintain its stable set of internal conditions. I want you to think about, how does it get that energy? That's what the next lecture is going to be about.